if we zoom in and look at what makes a cell, I think probably the most important single feature of life as we know it, in other words, life on Earth, is this structure, the lipid bilayer, right? So we talked a little bit about phospholipids the other day. Phospholipids are game-changing molecules because they have this region here, which is hydrophobic, and then their head region here is hydrophilic. So if we put water around, the water tends to go on the head side, and the tails end up sticking towards each other. Now, what makes that important is it gives us a zone of hydrophobic molecules. Now, what's special about that is ions and charged substances won't cross through that region because there's no electronegativity in here, right? So if you have a sodium ion or a potassium ion or a chloride ion or a bicarb or a calcium or a protein, long list here, that's the idea, those molecules cannot wiggle their way through from one side of the membrane to another because the region in the middle is hydrophobic. That is the key to the lipid bilayer. It creates an inside the cell, which we'll call intracellular, and an outside the cell, extracellular. Now, why is that difference so important? Because essentially that's what makes alive, is that the inside is different than the outside, right? So because of this structure, we get this exclusion of material. So the only way to get from the inside to the outside or vice versa, two causes. One, either be a molecule that doesn't have much electronegativity, right, like a lipid or a gas, like CO2 or oxygen, or have a protein transporter of some kind, a channel, some kind of a protein that allows crossing of a molecule from the outside to the inside. All right, so now that we have our barrier, we can look and see, well, how does the inside of a cell differ from the outside? Why is this inside-outside thing so important? The first two are pivotal, okay? And essentially, we're going to be talking about the difference between sodium and potassium concentrations for the next 2 days in class, because it's the first two lines there that establish what we call membrane potential, which is the foundation then for action potential, or how do cells rapidly adapt to their current environment. Okay, so in the extracellular fluid, there's a lot of sodium and very little potassium. In the intracellular fluid, that's reversed, okay? Now, the way I remember this, I told you I was a scuba diver, right? So, ocean water, primarily sodium is the main cation, okay? So cells evolved in the ocean. So the extracellular fluid, high sodium concentrations. Potassium, somewhere along the line in early living evolution, potassium became this rare and special mineral that got sequestered inside of membranes. So inside of cells, potassium is very high. Outside of cells, sodium is very high. Now, what that means is that the tendency for movement in those two ions is opposite, right? It means if you open a gate, sodium's going to go rushing in the cell, if it can. If you open a potassium gate, potassium's going to go rushing out. You see, so they go in opposite directions. We're going to use that to build membrane potential later this week. Okay, the next one down equally is important. Okay, look, there's very, very little calcium inside the cell. There's lots of calcium comparatively. Look at all those zeros, right? So we have a huge order of magnitude, even bigger order of magnitude difference than sodium and potassium with calcium. This is why calcium makes stuff happen. Because the concentration gradient is so big, when you open a calcium channel, you get a lot of calcium real fast, okay? Because of that phenomena, it means calcium is connected to our rapid responses. So like every time a neuron fires, that's calcium that's making that happen. Every time a muscle contracts, that's calcium. And this is the reason for it. We have this huge concentration gradient for calcium between the inside and the outside. Now, what I don't want you to think is that the inside of the cell minds having calcium. Calcium makes things happen. It's just calcium levels are kept very low because whenever calcium is present, it's triggering something to occur. So in a cell that's at rest, we'll have a very low calcium level, right? From there, the differences are less important. A few I am going to point out. Chloride, mostly on the outside of cells, okay? The negativity, you know that for every positive in a solution, there has to be a negative, otherwise we'd stick to the wall, so to speak, right? Well, inside the cell, most of our negatives, most of our anions are proteins, right? Because proteins carry a small negative charge. So it means that most of our chloride is found outside of cells, okay? Bicarbonate we're going to talk quite a bit about in the respiratory system. It's our primary buffer, bicarbonate and carbonic acid, that buffer system. Again, lots of it outside the cell, relatively less inside the cell, but you see not a huge difference there. Okay, and then glucose, you'll notice a big difference there. Glucose high outside, low inside. This one makes perfect sense. Glucose is fuel, and cells are always hungry, right? So if there's glucose inside a cell, it gets gobbled up as fuel. It doesn't hang around as glucose. So that makes the concentration of glucose inside of cells low all the time. Now, that gives us an added benefit. If we open a glucose channel, well, the concentration gradient is going to push glucose into cells, right? So as long as the body maintains a healthy blood glucose concentration, in other words, the glucose in the blood is high enough, all cells have to do is open the door and glucose will come in and fill the need, right? More on that in our metabolism chapter. Amino acids, flip side. There's much more amino acids inside of cells than outside. And that's because it's inside of cells that amino acids play their biggest role, right? Amino acids become proteins. Where are proteins built? Inside of cells. Okay, so that means that you have to have a higher concentration inside than outside. Okay, cholesterol, phospholipids, and fats, mostly found inside of cells. Okay, the reason why those are all non-water soluble substances. There's no place for them to be outside of cells, right? Because we're mostly water. And then we get down to the gases, PO2 and PCO2. Oxygen concentration inside of the cell, usually low compared to the outside. Why? Consumption, right? So it's inside the cell that oxygen is consumed by the mitochondria. So if there's oxygen around and the cell is busy, concentrations will get very low. Opposite for CO2. Where does CO2 come from? It comes from the mitochondria. Well, where are they? Inside of cells. So it shouldn't surprise you that the CO2 level is high inside of cells and low outside. And then finally, pH. pH is our kind of convoluted measure of hydrogen ion concentration, okay, our acidity, right? Lower is more acidic. Remember always, lower pH is more hydrogen ions. Don't let that confuse you from the beginning. So why are there more hydrogen ions inside than outside? Because we make acid. That's why. Our metabolism tends to spill hydrogen ions. So anywhere we have busy cells, we have hydrogen ion production, and we have a lower pH than in a non-living, non-busy part of the body, right? And then finally, proteins. Much more protein inside of cells than outside of cells because it's structure. Right? Protein is creating the cytoskeleton. Protein is creating the machinery of the cell. So we have much more inside than outside. So for a simple slide, simple table, there's actually quite a lot of physiology in this one relationship.